Hello, I'm Allie McDermott, and today I'm going to talk to you about DNA replication as well as the differences and similarities between prokaryotic and eukaryotic cells in DNA replication, the replication process, and DNA polymerases. DNA replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. The reason that the replication process is so important is because when a cell divides, the two new daughter cells must contain the same genetic information, or DNA, as the parent cell. Because DNA replication is such a crucial process, the cells have to ensure that no mistakes or mutations are introduced. So the cell proofreads the newly synthesized DNA to make sure no mutations have occurred. Once the cell has been replicated, it can start dividing into two cells, each containing the same genetic information as the original. DNA replication begins at specific origins of replication of the genome. This is where the unwinding of DNA begins to start new strands of DNA. The replication process begins with initiation. This process is started at the origins, which are targeted by initiator proteins. Once the origin is located, the initiators recruit other proteins and form the pre-replication complex, which unzips the double-stranded DNA. Next is elongation, and during this, a primer sequence is added with complementary RNA nucleotides, which are then replaced by DNA nucleotides. The rate of DNA replication in a living cell was first measured at the rate of T4 DNA elongation in phage-infected E. coli. The replication fork is a structure that forms within the nucleus during DNA replication. It is created by helicases, which break the hydrogen bonds holding the two DNA strands together. The resulting structure has two branches, each one made up of a single strand of DNA. These two strands serve as the template for the leading and lagging strands, which will be created by DNA polymerase. The leading strand is the strand of nascent DNA, which is being synthesized in the same direction as the growing replication fork. The lagging strand is the strand of nascent DNA whose direction of synthesis is opposite to the direction of the growing replication fork. Because of its orientation, replication of the lagging strand is more complicated as compared to that of the leading strand. As a consequence, the DNA polymerase on this strand is seen to lag behind the other strand. At the replication fork, many replication enzymes assemble on the DNA into a complex molecular machine. Replication machineries consist of factors involved in DNA replication. They include primosodors and replication enzymes, single-stranded DNA binding proteins. These replication machineries are called replisomes, or DNA replicase systems. The final step is termination. It requires that the progress of the DNA replication fork must be stopped or blocked. DNA polymerases are enzymes that create DNA molecules by assembling nucleotides, which are the foundation of DNA. Every time that a cell divides, it requires DNA polymerase to help duplicate the cell's DNA, so that the copy of the original DNA molecule can be passed down to each of the daughter cells. DNA polymerase adds a new strand of DNA by extending an existing nucleotide chain, adding new nucleotides to the template strand one at a time. DNA polymerase are known to have highly conserved structure, which means that they vary very little from species to species independent of their domain structures. The conserved structure indicate important irreplaceable functions of the cell, which provides evolutionary advantages. In general, DNA polymerases are highly accurate, with an intrinsic error rate of less than one mistake per every 10 to the 7th nucleotides added. Both prokaryotic and eukaryotic cells have DNA as their genetic material and are both membrane-bound. They both have ribosomes and are both amazingly diverse in forms. They contain ribosomes, cytoplasm, plasma membrane, vacuoles, and sometimes have cell walls. They also share a similar basic metabolism. They each have one strand and one old strand and one new strand in each daughter cell. The replication process is generally the same for both prokaryotic and eukaryotic cells. Both types of organisms also follow a pattern called semi-conservative replication. Both types of organisms begin new DNA strands with a small primer of RNA. Prokaryotic cells are unicellular organisms that lack organelles and other internal membrane-bound structures. These lack a nucleus but instead have a single chromosome of DNA. The simple structure is a good thing because it allows them to produce very quickly and effectively. Because the cell is simpler, it makes replication simpler. The DNA in prokaryotic cell is circular. Prokaryotes can be divided into two domains, archaea and bacteria. Bacteria possess protein-based bacterial microcompartments. DNA replication occurs inside the cytoplasm and has only one origin of replication, and it is formed by about 100 to 200 nucleotides. Only two replication forks are formed in each replicating prokaryotic chromosome. The initiation of DNA replication in prokaryotes is carried out by protein DNA A and DNA B. Prokaryotes also undergo binary fission. Most prokaryotic cells do not go through a well-defined cell cycle, but instead continuously copy their DNA. During rapid growth, this can also result in concurrent occurrence of multiple rounds of replication. Replication also happens at a much faster rate in prokaryotic cells than in eukaryotes. Some bacteria takes only 40 minutes to replicate, while it takes some eukaryotes around 400 hours. Eukaryotic cells are single-celled or multicellular organisms whose cells contain a distinct membrane-bound nucleus. They have cells that are organized into complex structures by internal membranes and a cytoskeleton. They also have linear DNA. DNA replication occurs in the nucleus with over 1,000 origins of replication. Each one is formed of about 150 nucleotides. It occurs at several points simultaneously in each chromosome. Several replication forks are formed in each replicating DNA. The DNA molecules have 50,000 or more replicons, but they do not occur at the same time. The initiation is carried out by multi-subunit proteins origin recognition complex. Replication is slow. Around only 100 nucleotides are added per second. Eukaryotes undergo mitosis. Within eukaryotes, DNA replication is controlled with the context of the cell cycle. As the cell grows and divides, it progresses through stages in the cell cycle. DNA replication takes place during the S phase, and the average eukaryotic cell has 25 times more DNA than a prokaryotic cell. Throughout this video, I have talked about DNA replication and the replication process, DNA polymerase, prokaryotic and eukaryotic cells, along with similarities that they share and their differences. I hope that this video has helped you learn a little bit more about DNA replication and all of the processes behind it. Thank you.